I started Thrive Bioscience, co-founded it 6 years ago in the Boston area because we found an urgent need in life sciences, which is to improve cell culture, stem cell culture, and tissue culture. I founded 11 companies, 4 of which have had multi-billion-dollar exits, and one of them was very similar to Thrive, where we automated cytology labs. Cytology labs read pap smears, and like cell culture had no tools, was done manually until we automated it. And now 85 to 90% of all pap smears are done using my instruments. We've changed the course of cervical cancer, and we sold the company for $6 billion. This is similar. Cell culture has almost no tools. Next slide, please. So cell culture is it's performed manually, even though it's really critical for bringing drugs to patients. We are the first company that is bringing needed analytics, automation, documentation, and high-quality images to cell biology. And as a result, we're producing for the first time data sets that can be used for machine learning, for data mining, and for breakthrough insights. Next slide, please. So our products are instruments with a very high content of software that allow researchers to more effectively study living cells. We have an experienced team, besides my own experience with many multi-billion-dollar exits. My co-founder is responsible for most of the sequencing instruments that are sold today. He built the instruments that Illumina sells. They actually acquired his company. Interestingly, most innovation by the large companies is bought from small companies like Thrive. So we have very significant collaborations. We have a collaboration with large pharma in Japan, developing an advanced 3D imaging system. And for each of the 7 software modules that we're developing, we have collaborations. So we have collaborations with the Center for Genomic Medicine at Mass General Hospital, Harvard Stem Cell Institute, Stanford Stem Cell Core, the Broad Institute, Wake Forest Institute for Regenerative Medicine, and other really leading institutions. There's very limited innovation in the field and therefore very limited competition. Since nobody has built these instruments before, we have built a very strong IP portfolio with 75 applications, patent applications, 24 of which have issued. So basically, somebody's going to have to buy our products or buy us. So we are just starting to have revenue. We expect $6 million in revenue in the coming year, 19 million after that, and in 5 years, growing to over 150 million, and being profitable in about 2 years. So we've raised $29 million, and most of our investors are from family offices. So the Estee Lauder family office, the Milstein family here in New York, Parker Brothers family. They sold Parker Brothers to General Mills, well known for their monopoly game. And they actually invest a lot in life sciences. So I'm actually spending monopoly money, which when I played the game as a kid, I always dreamed that money would be real. So we're spending some of the Parker Brothers money. Interestingly, they're very good life science investors because I didn't realize it, but those games are covered by patents. And a lot of their barrier to entry is based on patents. And they really understood our patent portfolio very well. So we're raising $8 million in a convertible note. The use of proceeds, we have an opportunity, if we could raise the remaining 4 million in the next few weeks, to acquire a company that is actually one of our suppliers, and it's the leading company in cell culture environmental controls. Reproduce and measure, and they control, and it's a fabulous combination. They expect to also do $6 million next year, $10 million the year after, and it advances us more quickly towards an exit or an IPO. So good science needs good cells, but we're not producing those good cells. So let me tell you about the current status of cell culture. So even though it's a 18 to $20 billion per year market, and is key to biomedical research, it is conducted almost entirely manually like it was 65 years ago. And let me explain to you how it's done. So cells are kept in incubators like the image on the upper right, and we take those cells out without our products and put them under a microscope, and researchers spend hours per day looking at millions of cells. And then they put the plate of cells back into the incubator, and guess what? All the data is lost. They don't remember those million cells. I would say that Uber knows more about us than we know about cells that we're studying for bringing medicines to people. There's a lot of stress that's put on these cells as they cross these environments, and as a result, the cells are highly variable and not comparable. So they vary across labs, across time, even researcher to researcher in the same lab. And amazingly, 14 to 30% of all cell lines are either mislabeled or contaminated. So what this means is a researcher thinks he's studying liver cells and screening drugs for liver cancer, but he actually has pancreatic cells. Or if he actually does have liver cells, it's been contaminated and has DNA from other cells. The cells are getting more and more distant from the patient, which is why we have cell culture. So we then take these cells of variable quality and poor quality, and we put them into instruments and run assays that we spend billions of dollars every year on. But it's really a garbage in, garbage out. Next slide, please. So this has resulted in what Nature magazine calls the crisis rocking research. This is known as the reproducibility crisis, reproducibility of research. And Amgen produced the first major study in this field. They studied 53 important milestone studies in oncology to see whether they could repeat them. They could only repeat 6 out of 53, which is incredible. And they spent 10 years trying to do this and had up to 100 scientists working on this. There's been no study that has shown that more than 50% of research is reproducible. So essentially, about half of the research spending in the US is wasted in the world. In the US, that's about $28 billion. And other studies have found that 50% of the reproducibility is the result of cell culture, which is what we're fixing. So this slows cures to patients. It also puts patients at risk because there are hundreds of thousands of patients who were enrolled in these clinical trials that are based on really irreproducible results, junk science. Next slide. So the solution is our instruments and software. There's a very high degree of software content. And we're providing imaging, analytics, automation, specifically to make research reproducible. And our instruments are enabling researchers to do things that they've never been able to do before. Rather than mine tell you how great the instrument is, one of our customers at University of Texas Medical Branch in Galveston, so they have a $20 million a year contract with Pfizer. Whenever you read about the variant and what the results of the coverage of their vaccine is, it's all done at University of Texas Medical Branch. It's the leading center in the United States in infectious disease. And the chairman of the biochemistry department, who has this Pfizer contract, actually said that Thrive will change the world. Thrive allows us to see and understand living dynamic cells and tissues in 3 dimensions over time with remarkable images. Essentially, our imaging and analytics is closer to biology. Next slide, please. So we have a family of instruments with increasing degrees of automation and analytics. We have 2 on the market. Most of our effort now is concentrated on software modules that we're developing with the collaborators that I mentioned. And it's very cross-disciplinary. We integrate imaging, robotics, analytics, fluidics, machine learning, databases, user experience, user interfaces. And we don't require FDA approval. Next slide, please. So a lot of the value and I guess secret sauce is in our imaging. And we've achieved the holy grail, which is that we can find a single cell in one of those plates, and no one else can. It's a needle in a haystack problem. And we're able to track it over time, which is incredibly valuable for things like CRISPR or single cell sorting. With genetic engineering, such as CRISPR, you end up with one or two or just a few cells, and you can't even find them, much less track them, which tells you a lot about cell health and data that you need. So we capture up to 60 focal planes, 2.5 to 50 microns apart. No other instrument does this. And we image all of the cells and all of the walls and all of the plates. So we build this massive database that's useful for documentation as well as for research insights. And it's not unusual for one of our scans to be 30,000 5-megapixel images. That's 90 gigabytes of data with each scan. Our sites are producing 2 to 3 terabytes a week.
I have a question.
Oh, sure.
So I have an important question. So you said there's no need for FDA approval, so there's no binary aspect to this investment.
That's right. So we're selling research tools which don't require FDA approval. We also are getting pulled very quickly into bioprocessing of drugs and therapeutics for the FDA, but we're not directly regulated. The entire process has to be GMP, good manufacturing process, but we don't have to be, as long as the overall process. So very little regulation.
And are you gonna get to later slides how your product is distributed, you know, what channels you use, who are the main buyers, does insurance buy it?
Sure, let me answer right away. I never believe in deferring a question. So we're using a direct sales model because it is a complex instrument. So we just hired our first 3 salespeople. Internationally, we use distribution. We offer it as both capital equipment and on a subscription basis. And most of the cost is actually our software, which is on a subscription basis. We don't have any involvement with insurance companies. This basically comes out of the research budgets of pharmaceutical companies mostly. So 2/3 of our market are pharmaceutical companies doing drug discovery and biotech companies basically doing drug discovery. And about 25% or so are basic researchers in mostly major institutions like the Broad Institute, Harvard Stem Cell Institute.
Can you give us a size of the market, like a market size?
Sure. So it's about $18 to $20 billion. Cell culture is done in 80,000 labs around the world. We actually, for the accessible market, serviceable accessible market, assume there's 20,000, only 1/4 of them, which I think is very conservative, which puts it at about a $10 billion market for just our instruments of the $20 billion or so that's being spent, and it's growing very quickly. It's the basis of all the drug discovery that's going on. So next slide, please. So to give you an example of the value of data, I mentioned to you the mislabeling rates, which is a result, by the way, of over time with manual processes. And data makes it all very clear, very simple. You can see that cells are behaving differently. So this was from the Broad Institute, and they noticed these outliers and took a look at them, and sure enough, they were mislabeled. Other institutions have had cases where cells were growing more slowly, and it turned out that the wrong reagent was used. Next slide, please. So hopefully a few pictures tell a lot of words. So this is why University of Texas Medical Branch said that Thrive will change the world. So current infectious disease research is only able to see a stained plant. So the standard way that viruses are studied is an overlay of viruses put on on a lawn of cells. And the virus eats away at the cells and creates plaques or holes. And nobody's been able to see those until they're stained. So nobody can see anything before, and once they're stained, they're dead, so nobody can see anything after. But we can. You don't need stains. In fact, you don't have to wait the 7 days. And there's a lot of data in the rate of change and in the way the plaque forms that has never been available before. And in fact, just by looking at these images, they discovered that when they were studying COVID-19 or SARS-CoV-2, they actually had a variant within their cell culture that was confounding the data. So for the first time, actually, drugs can be studied over time and not just at one point in time. Next slide, please. So this is a holy grail. This is what I mentioned before. Nobody's been able to find a single, Nobody's been able to find a single cell in a well. But here you can see the single cell doubling, and this is a stem cell, actually becomes 2 colonies, which is, they're monoclonal colonies, which is the gold standard in research. As I mentioned, this is incredibly important for CRISPR, for single cell sorting. In all of these, you have to start with good cells, and then you end up with a few cells that you have to grow. And looks like there's money. So the Broad Institute said that we're the best instrument for imaging tissues. And this is the fastest growing area in cell culture because currently you have adherent cells mostly that are sitting on the bottom of a plate, and then you have animals, and you have nothing in between. And so 3D cell culture is closer to the organ, to the animal, but it's still in the lab with a much lower cost. So that's why it's the fastest growing area, but nobody can image them nearly as well as we can. And that's a lot because of these stacks that we have, up to 60 focal planes. We can actually do more than that, but we had to pick a big number. I think Massachusetts General Hospital's done 120 stacks. Next slide, please. So in terms of our background, I mentioned my own, which my first 3 companies were artificial intelligence software companies. I was probably still the largest standalone AI company, which to this day, it was acquired by Fair Isaac, monitors 90% of all credit cards and debit cards for fraud using neural networks. We took the company public and we were acquired. And then since then, I've started 8 life science companies. My co-founder spent 25 years in genomics, developed the most important instrument probably existing now in life sciences, which are sequencers. There are 3 types. He developed 2 of them, and by far the largest is sold by Illumina, which they've acquired his company that made those. Our chairman was president of Thermo Fisher's Laboratory Products Group, the largest group in the world. We also have Brock Reeve, executive director of the Harvard Stem Cell Institute on our board, and one of our early customers and one of our largest markets.
Isn't Brock Christopher Reeve's brother?
Yes. Yeah, so Brock Reeve is Christopher Reeve, the Superman actor who had a spinal injury and died of that. Was having really good results with stem cell therapies. He died of other causes, but his brother Brock really dedicated the rest of his life to regenerative medicine. And in 2008, he took over Harvard Stem Cell Institute. It was like 4 people, and now it's several hundred. Next slide, please. So our financing history, as I mentioned, we've raised $29 million to date. We've had nice, consistent increases in our pre and post money valuation, pre-money going from 6 million to 55 million. We're going to hopefully close the convertible note so we can acquire this other company, profitable company in our field, also one of our suppliers. And we raised 4 million of the 8 million. I don't know if I'll be able to do 4 million by the end of the year, given Christmas and holidays. But we That convertible note converts into Series B at a 20% discount, and in Q2, we'll do the actual Series B, probably for $10 to $12 million. We actually have a term sheet for $10 million. It's not a life science investor, so I don't know if we're going to continue with this, but the valuation from that term sheet increases our pre-money, and we use that from $55 million to $80 million. Our shareholders include the family office, as I mentioned. We also have 7 angel groups, Life Science Angels in California, New York Angels, most of the Boston-based ones. I used to live in China. I ran a venture capital fund there. So we actually have a number of large families in companies who don't like to be mentioned. And a number of industry veterans, former CEOs of Novartis and Patheon and Bremer Bio and Exact Sciences. Next slide. So I'm not looking to sell this company, but one always has to be aware of it, or at least not sell it right now, because I think we're still really adding so much value, and it would be too early. But I always keep an eye on that. And this is an attractive area with very high multiples. And this area is imaging and live cell biology. It's one of the fastest growth areas in life sciences. The multiples for public companies are average about 30 to 35 times trailing 12-month revenues, and in M&A about 10 times trailing 12-month revenues. M&A is lower because you have more companies there that ran into problems. So using the low end of those multiples, we expect a 20x return. My average return on my 11 companies is about 30x. So I expect to do it again. I actually expect to do better. This is one of the largest markets that I've seen in life sciences. We have many companies that we fit into well. All of the imaging companies, the laboratory companies, so Agilent, Danaher, Nikon, Thermo, Lonza, PerkinElmer. And there are a lot of very good comparables as well for public companies at the valuations that I talked about. So that's my presentation. I don't know if we have time for any questions.
Time for a question or two? Tom, I'm going to tell you that you fit the profile of somebody that you should get invested in, because we've had multiple guys who are multi-exit, biosciences, healthcare tech founders, and they've all raised lots of money and been successful time after. So it's kind of like it's a proven path, and it's actually fantastic what you've done. One of the big barriers to investment is always this binary choice that you see in pharma, but you've avoided that.
Yes.
And I don't know, that was strategic up front?
It was, I couldn't live through it anymore. I ran several public specialty pharmaceutical companies, and I just couldn't live again with unblinding of phase 3 data, not knowing what's going to happen, and sometimes the data doesn't come out positively, even though you know that you have a drug that helps people. You just haven't been able to figure out the subpopulation. Those binary events are terrible and greatly increase the risk. So I rather sell tools to those people who are doing those studies.
Hey, Tom?
Yep.
What was use of funds?
So use of funds are sales and marketing, because we're just hiring our sales force. Mostly software engineering. The hardware has been extremely reliable, extremely flexible architecture. And then third, the acquisition of this company that is also in cell culture and profitable company.